Hi, Alex. How are you? Sorry, I'm just setting up right now. There we go. Let me get you the chat out. How is it going? Are you finished your mocks now? What exam board are you most familiar with? So the exam board that I'm most familiar with is OCR A and AQA. It's kind of one of the two of them, but OCR A, I know that spec off by heart, basically. Um, I am good, thank you. I'm good, thank you, Alex. But yeah, that's the one that I tend to do. Yeah, finished, got bio back, was 2 marks off an A. Oh, Alex, so close. But the best thing that you can do now is use that as a way to guide you on what do you need to do to move forward. So that's a really good place to be, and it's about how to move forward. Chidima, how are you? I saw you answer some questions on my videos from earlier. Oh, I've done that thing again that I always do. I don't know how I do this. There we go. What did you just say? I did the 2025 paper this year, but I fumbled it. Oh, I'm sorry to hear that. 5 hormones that maintain homeostasis. You want me to name 5 hormones that maintain homeostasis? Let's think this through. You've got insulin, you've got glucagon if we're talking about the blood vessels. No, we're not. What am I talking about? See, this is where my brain has to get engaged with it. Right, hormonal. If we're talking about glucose concentration, insulin and the glucagon. So insulin, we want to get our blood sugar levels to decrease. Glucagon, if we want our blood sugar levels to increase. You have got antidiuretic hormone if we are doing osmoregulation. Then we can bring in our aldosterone as well, because aldosterone is necessary for maintaining our sodium levels and our potassium levels. Nice, Chidima, that's really good. And then I would bring in the likes of thyroxine. It's not one that you have to know though for your exam board, but if we were looking at homeostasis, we'd be looking at things like our temperature regulation, and your thyroxine is necessary for the metabolic rate within the cells, and that would therefore link to that. However, if we're going to go down, potential cortisol would be one that you could name if we're looking at maintaining carbohydrates, potentially. Depends what the question's actually looking for. I doubt that they'd ask you to name that many for homeostasis. I need to deep analyze my performance and see how to improve. Absolutely, Alex, that's exactly what I would agree with. It's usually simple questions that I struggle on, like purpose of sampling, and I said to measure levels of biodiversity accurately, whereas I needed to say that it would be otherwise it would take too long. Yeah, so that's the key thing. It's not about focusing on what the exam board specifically for that mark scheme wanted. It's about what are you doing with your answers that's not giving you the marks. And that's one of the things that does come through quite often. The number of students who are generic with their answers, and they think that that's going to get them the marking point, and they need to be a bit more specific. So definitely identifying what it is that you need to put in there. Hi. Right, let me go back and just double check if there's any questions here. You're stuck on a C, but I know the content really well. Any tips? Practice exam questions from other exam boards as well, because that will help you moving forward. As long as you make sure it's only on your exam specific content. Chidima, good, you are finally understanding muscles. Yay! I just finished. I was super excited. I just finished. Oh, you're back on here now. Just finished the muscles. Oh my god, I just saw you got your biology offer for Oxford. That's amazing. Congratulations. That's so positive. Well done. Well done. It's really heartbreaking when you get a rejection. So for those of you who have got rejected or didn't get an offer. I'm sorry to hear that. But for those of you who have, absolutely celebrate. I'm so proud of you. I had the same question. I think I wasn't specific enough. Oh, bless you. It's hard. Cloning's a different story. We can look at some cloning. Right. Have we got any more questions on here? Or am I good to go now? I'm sorry. I did say that because I knew you were coming. Oh, thank you. Do you know what? It'd be amazing if you did support me. Hi, Callum. Oh, first uni exam done. Amazing. How'd it go? How was it? Managed to answer last time. Oh, I'm really pleased that helped you. Pramila, yeah, do you know, it's a funny one. I actually started drawing myelin sheath stuff because I was thinking about how I haven't done my neuronal communications workbook yet and study guide. Got hungry halfway through. That's never good. Never good when you get hungry in an exam. When we purchase it, when you purchase it, automatically you'll get sent a link to it. So as soon as you've purchased it, you get a link for you to download it. It's been 1 week since I applied for uni and I haven't got any offers. Is that bad? No, it's not bad. You just got to wait. It's going to take time. They're going to put lots of things out there. Every university is very different, so unfortunately you've got to just wait on that one. I am so happy for you too. Shush, Alex. That 6 marker on the muscle. It's an interesting one because a lot of students will go, oh, there's a 6 marker in 2025's exam on smooth cardiac muscles. And so therefore they'll believe that the muscles won't come up again. And it's about knowing what the exam board are going to do and how they're going to do it. My first offer took roughly 3 weeks, so it takes time. So we had a first offer took 3 weeks after the interview, so it is about taking stock and knowing that it will take time. Do I do biology? Absolutely. Do I only do biology? With A-level, yes. GCSE, I can do all 3, but I don't have that much time at the moment to offer GCSE, so I'm trying to do a bit more, but I have to factor in the clients that I've got and what they need. But yes, I can do the others. You could have 2 rejections. That makes me so sad. So Christine, I thought people would have been finished. It's an interesting one. Did you have one on the T-test? Every exam paper they will give you a statistical analysis. Oh, Zara, good luck. Definitely check out my YouTube channel, Zara. I have got videos more linked at prep for paper 1. I haven't done paper 2 yet. If I get a bit of time, I will do. But if you're AQA, go and look at my combined science, AQA, biology, and my combined science, chemistry. I've got some videos out there for you that may help. Oh, it's so hard. No T-test, just Simpson Index. It's an interesting one because the exam papers and the questions, if your teachers are good at what they should be doing, they should be mixing up those questions slightly or adapting them, but sometimes we just don't have the time. It's quite difficult. So we do sometimes just give you exactly the same paper, but ideally they would be switching it around and giving you different questions. Oh, that's really lovely, Pramila. Thank you. It's lovely to be appreciated. What subjects are best to choose in college? That really comes down to what you're interested in, because A-levels are hard and you are going to have to really focus on pushing yourself through the bad parts of the subject that you really don't enjoy. Ask any of the A-level students who are here now. They'll tell you it's not easy doing biology. It's amazing. It's such a nice... I love it. This is why I do it. But it's a lovely subject when you really can understand and see biology in action. But it's so intricate and the detail you have to go into makes it quite challenging. So that can be quite difficult. You got 54. That's amazing. And the fact it's only two marks off an 8, honestly, that's a really high grade. A lot of students, interestingly, did really bad at the paper 3 on the 2025 exams. And so I would say it's it depends each year which one's the harder one, and which ones that you should work on. But it's so tough to know. It's so tough to know. Right, should we do some actual learning tonight? I feel like we should do some actual learning, because I feel like it would help you guys out. Mocks are done now. You can't do anything about them. Grade 7 is really good in science. Ideally, keep going. You can push that up. If you're getting a grade 7 now, then you should be able to make your way towards an 8. So that's what I would say. If you're getting 7s now, work towards the 8, and you never know, you could get the 9. But definitely get a solid foundation of the content, because if you're going to choose it for A level, you definitely need it, because everything you do in GCSE goes into more details when you get into the A level. We should get the article soon, right? Two weeks before, so probably Friday. Oh, interesting. Right. Here we go. What are we going to do tonight, guys? What do you want to actually cover? Or are you just on here to have a chat with everybody? It's deadline day. Deadline day. For those of you who aren't early entries, so people are stressing. This is for our Queen Mary. And oh, that makes sense. What do you get? So you get an article that you have to read, and then they ask you questions based on your understanding of the article that you've read. I read an interesting article. So one of my friends is for 10 minutes they ask questions. Oh, okay. So you get the article to read beforehand, and then after you've read the article, you can then analyze it, and then you go to the interview, but you don't know what they're going to ask you in interview. Is that how it works? That's very much what it used to be like with the old style A-level biology exams, where you would actually be given an article, and as a teacher, we would go through and annotate with you all the things that we think that they could ask you based on this article. And then it would, once you've got that, you would then basically sit the exam and see what questions they asked you. It was great. I love that kind of thing. Maybe I'll see you. You're the second person I know on that day. Oh, that's so cool. If you guys actually end up meeting at interview. Talking about interviews, when I went to my Cambridge interview, I was there with this girl, and I still remember the day of the interview. I rocked on up to the interview, so nervous, hadn't done any prep for it because I hadn't got the interview information in time. And I rocked on up, and this girl was sitting in the chair, and there was just two of us to begin with. And all I kept thinking when I saw her is, I was like, wow, she has got a massive smile and like so many teeth inside her mouth. And that was my first thing. And then she started talking to me, and I was like, I really like you. You're really cool. You are awesome. And then it was a case of, I was like, okay, we're kind of vibing here. This is really cool. And to this day, we are still best friends. And she was the person who I sat beside and interviewed with at Cambridge. And we both have, so on my 30th, for my 30th birthday, she'd actually gone out to America. Because I introduced her to her husband in Cambridge. I introduced her husband, who was American. They went out there for work, and for my 30th birthday, I went out to America to visit them, and now they're back here. And yeah, it's awesome. And we're both biology teachers, obviously, because we went to Cambridge together to do our postgrad. And yeah, it was really awesome that on our interview day. Oh, thank you. Whoever's just bought my one of my study guides, I just got a notification. Ah, thank you. That makes me really happy. I hope it is the muscle one. You get lots out of it, or any of them, to be honest, because I'm working so hard on them. I'm really struggling getting the grades I need for college. I'm currently working at 3. Okay, so if you're working at a 3, that means that you're doing GCSE. So what I would say is lots and lots of practice papers will help you, but definitely check out my videos for my GCSE stuff and see if that helps us. Also, science shorts, he's got lots of good videos out there for you, so do check that out. That will also help. But it for GCSE, it's definitely about repetition of the content. Tassima is quite a good app that you can use as well if it's still going. But I don't know if you how much you have to pay for that, to be honest. But with GCSE, there is so much to learn at that level, but it's very much you just memorize and get it across. If you're wanting to get up to a grade 5, if you want to go up to grade 9, then you need the knowledge. So it's not just about memorization. So if you're trying to get up from a 3 towards a 5, definitely just repetition, repetition, repetition. So yeah, guys, if you have any questions specific about the content and you want me to cover it, then please do let me know. Chidama, you mentioned cloning, that you're struggling with cloning. Do you want me to cover a bit on cloning, or are you just having a chat about, happy with the chatting about the whole interviews and different stuff? Okay, last year I got a 7 all across. I'm not really too sure what happened. Oh, okay. So if you went from a 7 to a 3, it could just be that you had a bad set of exams. And so it's about identifying where you went wrong and what you can do to improve it. But definitely, if we're hitting 7s and now getting 3s, it would be that you just need to go back over the content. Some of it's really tough. Some would be really easy, and then some will just be really tough for you. But for GCSE, it's definitely about repetition as much as you can. Oh, please just distract you. Good. If you can go through cloning, absolutely can go through cloning. 100% I can do that. Chidama is so hard because right now it is the biggest focus, and it's what everybody's talking about, and then you're comparing yourself to other people. Please try not to compare yourself to other people, because that's not going to help you. What is going to help you is thinking, where and what is it that you want to do, and how can you then use that to move forward. Things happen, and these things that happen are there to get us to reconsider and rethink about where and what we do. As I've said to you many times before, all of you who have been here and listened to me drone on about my life, I was the student who hated school. I was a student who dropped out at the end of year 12 because school wasn't for me, and I was always failing at things. I couldn't do as well as anyone else. I just struggled. I didn't like it. And I just needed a bit more time. And I've just seen, Hibbs has just jumped on here saying, I failed my first year of A-level biology, but I retook and got an A. Just needed to look in. Absolutely. Sometimes you just need to take your basically put your blinkers on, not take them off, put your blinkers on and be like, this is what I'm doing, and this is all that I'm doing, this is only what I'm doing. And if I think about all these other things, I get so distracted I can't succeed. And that's what you need to do, is try and push past those rejections. They're done, they're dusted, you can't change them. Move forward. It's not going to define you. All it's going to do is be that, oh well, I got rejected. In like 5-10 years time, you'll be like, well, I got rejected from there, because I did. Like 20 years ago, I got rejected from Oxford. I didn't even get close to an interview from Oxford. They rejected me within a second of me putting that down. I heard your book clip but didn't put my email in. How can you access it? Hold on, Hannah, let me have a look. Let me have a look. I applied to do my PGC at Oxford and Cambridge. So when you do your PGC, which is your postgrad certificate in education, so when you do that, you basically can apply to 4 universities, or you could 20 years ago. And hold on, I'm just looking up Hannah, who just said she ordered it but it didn't come through, didn't put your email address in. Hannah, can you send me an email with your full name? and I can check, because I'm guessing, was it the nucleotides one that you bought? I found someone who spells their name different to you, so I'm just double checking I've got the right one. So if you can send me an email, it was module 1. When did you order it? Just now. Oh, hold on. That, are you sure it's definitely gone through? Because I haven't had it come through. I've just had one person pay for one today, and that isn't a name that is yours. You sure it went through? Hold on. Let me see if there's one pending. No orders pending. Can you still send me an email? Send me a screenshot of the notification from the Barclay card that it came through from the Squarespace. If you send that to my email address to show it, but it's looking like it's not gone through on my end, potentially because you don't have a correct email address. But double check. But send me an email. If you send me an email with the notification to say that it's all gone through, and then I'll be able to sort that out for you. But on the part, it's not showing. You would have been my 81st customer, which would be lovely. Thank you for that. I appreciate it. Right, I've lost you guys. Where have you gone? Sorry, I'm chatting away here. Chatting away. Right. How do you manage the volume of content? What I would say about now, if you're year 13, you're still learning lots of new content, and it can feel quite daunting. So how do I manage the amount of content? I would say to manage the amount of content that you've got, it's about you taking the time to look at the stuff that you hate, and the other stuff will fall into place. So I'm going to show you what I mean. So I am going to do cloning now, guys. I know I've done a bit of a chatting away, but I want to do some actual teaching on these sessions. So with regards to cloning, and we'll start with the animal cloning. Oh, perfect. It's come through. Oh, you've now got it. Amazing, Hannah. I'm so pleased. And thank you. Thank you for actually purchasing. That hasn't come through on my end though yet. So that's interesting. So thank you. I appreciate your purchase. This is what allows me to be able to do more, and obviously I want to do more for you guys. Right, Chidima is listening. We're going to do some cloning. We're going to do some animal cloning, artificial embryo twinning. I had to laugh actually because I was doing this with one of my one-to-one students the other day there, and I was laughing about how bad my diagrams are. I need to improve them, and these ones are good, but when we get to my sheet ones, I'm like, oh, what was I doing? It's really funny. But let's move on and let's keep going. So when we are looking at artificial twinning, so we're looking at embryo twinning. So my mum is an identical twin. And because my mum is an identical twin, that therefore means that she is genetically identical to my aunt. Now, when we are looking at natural embryo twinning, that is where identical twins come from. So there was one egg, one sperm that fertilized to produce the zygote, the fertilized egg cell. So we have two diploid, so two sets of every chromosome, so diploid cells that were reduced down to haploid cells through the process of meiosis. So someone said, how do you cover all the content? So right now I'm talking about cloning, but I'm also bringing in your inheritance and your number of chromosomes. I'm going to talk about cell structure. I'm going to talk about the cells and how they divide their nucleus through the process of either mitotic division or meiotic division. So this is where you're actually revising lots of topic areas, but under the bracket of cloning. So meiosis is going to occur, and when meiosis occurs, we're going to produce haploid cells. So fertilization, when we're talking about artificial twinning of these embryos, what we've got is fertilization has occurred. Now that could be natural or through the process of IVF. So we have this fertilization process. these fertilized egg cells, zygotes, which have now got diploid nuclei, and because they've got diploid nuclei, they're going to go through this mitotic division. So when they go through mitotic division, they're going to go from 1 cell to 2 cells, 2 cells to 4, 4 cells to 8, 8 cells to 16, and so forth. So we're going to end up with these multicellular embryos. Now, it's really important that you understand that cells that are in the early stages, when they're embryonic, if they're in the early stages of the embryo up to the 16 cell stage, they are totipotent. So that's going back to module 2. They are totipotent, which means they have the potency to become any cell type as well as the whole organism. So if we take these zygotes and we allow them to divide by mitosis to form embryos, but we catch them at the cell stage where they have yet to actually become pluripotent, what we can then do is select only one, the one that we want with the desired characteristics, so the desired phenotypes that we're looking for. And what we would then do is we would actually split that embryo up into several smaller genetically identical embryos. And as I said before, it's about them being totipotent. So the fact that they're totipotent means that they can go through that division. If your mother's an identical twin, does that mean, actually, I don't know what it means, somebody's embryo split in half? Yeah, my gran's. So my gran had 2 daughters, and her and my grandpa thought to themselves, you know what, we're going to have a 3rd child. And so they were like, yep, we're going to have a 3rd child. This is going to be amazing. They obviously got pregnant, and then lo and behold, that embryo actually split inside the oviduct, the tubes that lead into the uterus, and both of the embryos implant. And because both of the embryos had implanted, what that then meant is they actually grew their own placenta and they grew their own amniotic sacs. And so basically they were one that divided into 2. And they therefore were born, and my mum and my Aunt Barbara look so alike, to the point that as children we would go up to them and be like, Mum, and then it would be a case of my Aunt Barbara turn around and be like, what are you talking about? Because she sounded so much deeper than my mum. We'd be like, oh, you're not my mum. And supposedly my Aunt Barbara would like lift us and pick us up as babies, and we'd be happy as Larry in her arms. And then the minute she spoke to us, we'd be like, oh, and we'd start crying. We'd be like, who is this strange woman? Now you can tell the difference between them now, and that is where the environment has an effect. So they are genetically identical, but the environment has an effect on the phenotype. But also you have to remember, going all the way back to DNA replication, when your DNA goes through DNA replication, you will have spontaneous mutations occur. So there will be slight differences that will have occurred in the cells over the years that make them be not exactly completely identical anymore. However, when they started out, they were. So anyway, back to this. So we have these embryos, and the embryos that are going to split. And then what we're going to do is we're going to take the embryos and we are going to insert them into a foster mother's uterus. Now, the uterus is basically just the house for the embryo to grow. So the embryo gets inserted into the foster mother's uterus, and hopefully, fingers crossed, hopefully that embryo implants into the lining of the uterus. Now, if the embryo doesn't implant into the lining of the uterus, there's not going to be this development of a placenta, which results in this exchange system from the mother to the fetus, allowing for the required oxygen and the removal of waste substances. So what you have is you have the fact that although we can try to insert the embryos into the uterus, one of the downsides, one of the disadvantages to cloning is that it's not always successful. So Chidama, you said, how do you split the embryos naturally? So naturally is that they will split on their own whilst going through that development and that division. And if you're going to do that in a lab, in a petri dish, you will therefore be able to basically suck the cells away and separate them that way. So it's, I mean, that's very simplified, but it's very much a process that can be done under the supervision and looking down a microscope, and it's like a really intricate process that needs to be done within sterile environment. However, naturally, if the embryo is going to divide on its own, it will needs to do it at a very early stages, and when it doesn't do that, that's what results in conjoined twins. And sometimes what can happen is the cell can go through this process, the embryo can go through this process where it's about to divide and produce twins, but then one of them gets sucked back in to the original embryo, and it is a really weird like process that occurs, and I don't know like enough detail to tell you exactly what's happening. But if you have, so sometimes some people actually don't know that they're twins, or some twins have actually engulfed and digested their twin without knowing whilst the development is going on. So it's that whole thing of biology is amazing because all these things are happening, but we're still figuring it out, and we don't actually really know it all, and we're still learning things, and we have to put more and more money and research into figuring out. And every time we learn something new, things that we thought in the past actually are no longer relevant. And that therefore means that we have to move forward with science. We have to advance with our knowledge. And with the opening of AI and the fact that we have this ability to use that to our advantage means that we're going to see massive improvements in a much shorter amount of time with this science and technology than we have done in the past. Because if you think about all these things, that where did these ideas come from? They came from someone going, I wonder if, but they had to do like so much research, more research than now they would have to do, that they have access to the internet where you can basically, the AI, I mean, it's just amazing what AI can do in the fact that it can actually read and go through all the different journals that are out there. But big thing here, although AI can help with the advancement of science in the fact that it stops us needing to spend hours reading every single journal, it can summarize things down. The problem is, if we rely on that summary, if we rely on just what the AI tells us and we don't question it, and we don't use our scientific know-how to actually say, well, hold on a minute, that doesn't actually make sense. That doesn't sound right. And that's one of the problems that we have with the use of AI and the way that students are using it, is they just believe everything that they're being told by the AI. And so what you need to be able to do is you need to be able to use your inquisitiveness and your critical thinking skills to actually say, hold on a minute, is that right? And I think students don't feel confident doing that. And it goes back to actually your interview. Make things up. Sorry, absolutely, Callum. ChatGPT does make things up, and you have to be really careful in what you're believing, basically. So and I'll use my daughter as an example for this. I keep using my daughter as an example. So I was driving her to swimming yesterday, and she was telling me about the fact that two of her classmates had seen a ghostly figure in the toilets, right? And there is method to my madness here. So they'd seen this ghostly figure in the toilet, and they had gone out and they told all the students in the class, there was this ghostly figure, and so my daughter comes out, she's like, Mummy, it's haunted, the girls' toilets is haunted. Okay, she's 6 years of age. And I said, what do you mean it's haunted? She tells me, these girls have seen the ghostly figure in the toilet. So I'm like, okay. So she's like, yeah, it's definitely true. It's true, Mummy, because they told me. And they didn't smile when they told me. And I was like, they didn't smile when they told you, so it must be true. What do you mean by that? She's like, well, you say you can tell when I'm lying because I smile. And I was like, right, okay, that's your telltale sign. So my daughter has a telltale sign that when she's trying to tell me a lie, she just gets this massive smile on her face, and I know straight away she's lying to me. And I was like, but that's not someone else's telltale sign. So she believed that because that was her telltale sign, I could tell when she was lying, because I know her, that meant that anyone who tells a story when they're telling and they're smiling are potentially lying. So I was like, oh, I need to educate you here. But on that one, that's what I think of when students do and use ChatGPT, that when they use ChatGPT, that they just take what something else has said to them and believe that as being the true answer, and they then go and repeat it. And again, like something else happened, said, oh, well, so and so definitely didn't do this because they said this. And I'm like, well, did you see it though? Do you know it? So you need to always be questioning so that you can actually figure out the truth, whether that be through using AI or whether that be through talking to different people. And scientists can lie too. So there are scientists out there that are forging data so that they can become published. And that is where you would try and repeat an investigation to see if you could get the same results. And if you can't get the same results, that should make you question and evaluate whether or not what they've done is correct, or have they got enough of a sample size? Have they done enough repeats, etc.? Alex, my cell division test for OCR is tomorrow. What should I do? You've got 2.5 hours tomorrow morning as well. Alex, if you haven't checked out my YouTube channel, I have a whole playlist basically on that topic area. Definitely check that out. That will help you potentially. But also, yeah, just relax and see how you get on in the actual test. The tests are a snapshot of how you are right now, and they are a way of you identifying what you don't know. So using that is really important. Oh, you've watched that. Okay, so then I would say go to PMT and use their practice questions they've done it in topic areas, so go and do practice questions. But if you've done it all and you know it all, then just use that as being right now I'm going to assess how much I get on that. Sometimes actually having the break is better than just forcing yourself to go through it. Anyway, back to this. Okay, so embryos inserted into the foster mother's uterus, but potentially might not actually be successful. If they are successful, we should end up with identically cloned offspring. So they will be identical to each other, not to the parents. And this is a tricky one. They are not identical to the parents because they have actually inherited different genes from each of the parents. They have inherited those alleles. Those alleles could be different to what the parent had. So they are genetically identical to each other because they were once one embryo that was then split into these smaller embryos. So that's one way in which we look at the artificial way that we can do cloning, so the embryo twinning. The other one, which actually is one that comes up quite often, is somatic cell nuclear transfer, SCNT. So somatic cell nuclear transfer, and this is where my little sheep come in that I need to redo my sheep. These were some of the diagrams that I did back when I was first starting out with Illustrator. It does make me chuckle now. So if I have adult A, I would therefore remove a somatic cell from it. Now, again, using your knowledge, it's a diploid cell. It is 2N, it has 2 sets of every chromosome. These chromosomes are contained within a nucleus. They're contained within a nucleus because I'm talking about a eukaryotic organism, and that's one of the distinguishing features. So this is module 2, revising as well. What else do we know? Well, we know that they are eukaryotic, so the DNA is associated with histone proteins. And so, again, using this topic area to recap module 2, the chromosomes are linear because we're talking about eukaryotic organisms. So then we know that what we're going to do is we're going to remove the nucleus from the somatic cell. We want to clone A. We want the genetic information that resulted in our sheep, in this case, which was A. So we want genetically identical offspring of A. What we're going to do is we're going to use a different sheep, in this case female B, and we are going to remove a gamete, an ovum, which we know through the process of meiosis, it is going to have a reduction division. So if you remember, mitosis and meiosis are prophase, metaphase, anaphase, telophase. When we're doing mitosis, it's PMAT. When we're doing meiosis, it's PMAT1 and PMAT2. Prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 2, metaphase 2, anaphase 2, telophase 2. So mitosis, we're going to do PMAT once and get genetically identical daughter cells. Meiosis, we're going to do PMAT twice, a reduction division. And what's important to note is during meiosis, our anaphase 1 is where our homologous chromosomes are separated. Our anaphase 2 is where our sister chromatids are separated. So we're going to remove the nucleus. We're going to remove this haploid nucleus, and that is called enucleated. So the ovum, the egg cell, has been enucleated because we have removed the nucleus from it. So because we've removed the nucleus, we now have an egg cell which can actually intake this nucleus that we removed from cell A. Now, we do what's known as electrofusion. We are going to give an electrical shock, and that is going to ensure that this egg cell will now take up this nucleus with the chromosomes from sheep A. And by giving it a electrical shock, that is going to activate it as if the egg and the sperm have actually fused together, and that has resulted in the nuclei fusing together, and we now have a zygote equivalent. So by giving this electrical shock, this is going to activate this egg cell to now go through that mitotic division, prophase, metaphase, anaphase, telophase, and that will result in us getting genetically identical daughter cells. Now, a lot of times students forget that mitosis is only part of the cell cycle. The cell has to go through interphase where it has G1, S phase, G2. So G1, protein synthesis is occurring. S phase, DNA replication is occurring, and that DNA replication is semi-conservative. So the DNA has been replicated, the cells are then going to, or the cell is then going to go through mitotic division to divide the nucleus so that we can result in having 2 genetically identical daughter cells after cytokinesis. So all these parts of the module 2 still could be assessed in the topic area to do with somatic cell nuclear transfer. Chidima, you've jumped over here to ask a question. Pick up Pima. Wait, what do you mean by adult? Is it male or female? And why does the germline cell come from a female? So the germline cell that we're going to use comes from a female because we are going to remove the nucleus when we enucleate it. If you think about it, it's an interesting one. When you have an egg cell and a sperm cell come together, the sperm cell is much smaller than the egg cell. The egg cell contains the nutrients and the resources needed for the cell to continue and grow. So the sperm cell, what actually happens, not that you guys need to know this, but if you're doing the IB, you do need to know this. When the sperm cell and the egg cell membranes fuse together, what that then does is that then allows for the nucleus inside the sperm cell to actually move through into the egg cell, and the rest of the sperm cell actually gets digested down and destroyed. So the only thing that's actually required after the fertilization process has occurred is that nucleus coming in. So by the fact that we're going to use the ovum from the female is because that's got all the necessary parts for the cell to go through the process of mitotic division. Because you've got to do, for G1, what's happening during G1 is the growth of the cell, but also the replication of the organelles, as well as the proteins which are needed for DNA replication. Does that make sense? Yeah, YouTube, there's no messages coming through on the live chat. So I'm guessing that's why nobody's writing anything on the YouTube anymore. Don't ever apologize when you ask questions. I love that. Ask you any questions. Right, I'm going to finish this off for you. And then, same as what we did before, we're going to insert that embryo is going to be transferred into the surrogate, and then the surrogate, the lamb, should end up being a clone of A. But what's really important is you need to understand. Oh, Alex is back. Nice, Alex. You need to understand that the mitochondrial DNA, which was inside the ovum, would actually be from B. So it's not just about A, it's about understanding that there will also be DNA which is different from the adult A that you've taken it from because of the mitochondrial DNA. So they've done that in a question to do with frogs before, where they were looking at different frogs, whether they were albino frogs, whether they were not, and they wanted you to identify that not all the DNA would have come from A. Some of it would have come from the donor of the ovum because of that mitochondrial DNA. And that is your animal cloning, the two types. Now, somatic cell nuclear transfer came up. I shouldn't have given that away in case you've not actually done your mock yet. In this year's exam, but still, again, it's one of those things that I would say, just because it has come up in a year like 2025 doesn't mean they're not going to assess it in 2026. In 2022, 2023 and 2024, they assessed different areas of the excretion module. And so therefore, what you have to understand is if they ask a question and they look at one part of, let's say, cloning, it doesn't mean they won't assess cloning again. It just means they'll look at a different part of cloning. So they might look at cloning and look, for example, at mitosis or meiosis in the production of the cells like I've just described, rather than it being the process of somatic cell nuclear transfer. Or the other thing that they've done in the past, and this is something that's coming through more and more, is that students are memorizing this content. They're memorizing enucleation, nucleus removed, electrofusion, embryo develops, input into the surrogate, and they're like, oh yeah, I know this topic area. And then they give you a set of results and they say that scientists decided to do this and they were successful on this one, but they weren't successful on that one, and they did this on that one. Evaluate the results that you've been given, conclude on the results you've been given, and that's where the students struggle the most, because they aren't confident
one, it taking the time to read the question, but two, they're also not confident in deciphering what the question's asking. So lots of practice on that is going to help you moving forward, I would say, when it comes to questions like this. Oh, you are very welcome, Em. I'm glad you're finding it helpful. These are the things I do these every Wednesday, because I know not everyone can afford private tuition. So for those of you who are new to my channel, I have my channel where I have the whole course is there for free for you in small videos. I'm currently building my intense revision course at the moment, so that's long form, more with questions and stuff. But for those of you who are looking for tutoring, I do group tuition sessions on a Monday and a Wednesday, depending on year 12 or year 13. But as I say, I'm not sure everyone can do this. I'm in year 13, so I'm definitely be using this. There you go. Whole thing. How much is the group tuition sessions? Are £25 for the hour. That's an hour you get with me where I've planned the lesson. So next week we're doing manipulating genome. So I plan the lesson. You get a notebook where I put questions in there for you to answer. And then what I do is throughout the session, as you're answering them, I give you like information on the content. And then I actually guide you on your answers, and I give you feedback as well. Then you get a copy of all my resources at the end with a copy of the link to the recording as well. So you can use that any other point when you're doing your own revision at another time. So that is currently what I do for my group tuition session. So I do year 13 on a Monday, and there are a maximum of 9 spaces available. So currently there are 5/4/5, depending on the weeks, in my current group tuition session. In my year 12 one that I run on a Wednesday, there are 2 students in that one. Same, it will only be maximum up to 9 students, because I need to be able to give you the guidance and the feedback that will help you. So it is a very individualized as much as I can, but I plan it to the nth degree, basically. And so yeah, that's my group tuition sessions that I do, but my one-to-one sessions are different, and I do that for all the exam boards. So if you're looking for one-to-one, then that I do during the day though, and that's based around timetables of when people are available. You are very welcome. You are very welcome. But I do the lives on a Wednesday for free, and if I've got some time, I sometimes do jump on and do lives on a Monday after I've done my group sessions, but it really just depends. One, how tired I am, and two, if I have something else that I am busy doing. But my next set of things, so I've done the muscles now. I need to finish the neuronal communications workbook. It's going to be the next one that I do, because I want to get the whole pack done, but I obviously have to do them in between the different sessions that I do. So my study guides are coming. I had hoped that I would have them done, but we've had lots of things happening. We had a tooth come out today. For those of you who were following when I was talking about my youngest having her shark teeth, one of the front ones has now fallen out today. So she was super excited. Bless my student that I had. They were banging on the window, and she popped in to show us that her tooth had popped out. But yeah, guys, I am going to finish. Thank you so much for joining. My skill hasn't covered module 5 or 6. We're going through them at the same time with 2 teachers, so it's quite a lot. It will feel like quite a lot, but it's a common theme because of how and what you need to do. I would say that you should, hopefully your teachers, when I was teaching in the schools, last time I was in the schools helping and supporting, I finished the whole course by the first week in May. So that should be whereabouts you are. Ideally, as a teacher, you want to finish by Easter, but sometimes
you have to wait because there's like other things going on, etc. So by the first week in May, that coursework, that whole course should be absolutely complete. So it's not unheard of that you are going through the content still now, and different teachers teach in different orders, so it really just depends. But if that's the case, then hopefully my channel should help you out. Which part of nerve impulse conduction are you struggling with? Are you struggling with the propagation, or are you struggling with the saltatory conduction? Which part are you struggling with the most? Have mocks at the end of January. Would you recommend focusing on older content now? No. So, no and yes, actually. That's a... Focus on module 2. Focus on nucleic acids and nucleotides, biological membranes, and cell diversity, so mitosis, meiosis. Actually, not anything about neurons. You haven't covered it yet. That's fine. That's lots of students haven't covered it. You've still got time. Don't worry about that. Don't stress yourself out about you haven't covered neurons. What you need to do is worry about the content you have been taught now and what you don't know of that content that you have been taught, and work your way from that. Note it down. So yeah, which bit? To be honest, I forget half the things I say. So if you ask the question again, I'll say something different. Saltatory conduction. Basically, all you need to know about saltatory conduction is that the action potential does not need to go all the way down every single channel in the axon. Instead, it only needs to go at the places where the node of Ranvier is, where there's no myelin sheath. And because of that, because it's hitting less channels, because less proteins are needed to activate the membrane, what that therefore means is that the impulse, the action potential, actually jumps from node to node. So saltatory conduction is just that. The action potential moves from node to node, which makes it go at a faster rate down the axon. So it means it can travel a lot further at a faster rate. What should I focus on now till you're... Oh, okay. What should you focus on now would be nucleic acid and nucleotides, because that does DNA replication, protein synthesis, mitotic division and meiosis, biological membranes. And although, like, biological molecules is really important, I don't feel like biological molecules is one that students don't put effort into. So although it's necessary, I don't feel like you need to revise that. No, so it's not the same in... So imagine, right, okay, this is where I'm going to do something really bizarre, and this should... I don't know if it will actually work. So imagine, which way have I gone in the thing? Got to see where I am on my camera now because I've got so many different... Right, okay, I'm here. So now imagine we've got depolarization, repolarization. So I'm just going to do D-re, okay? So depolarization, repolarization, hyperpolarization. Then you've got depolarization, repolarization, hyperpolarization. Depolarization, repolarization, hyperpolarization. And I'm going to keep going, right? So starting over here. I'm doing this, 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 right? Now, if I'm doing saltatory conduction, I do this, and then I'm straight over here. So there's no waiting for the movement. I'm able to go from here to here straight away. So because of that, that is going to make it much quicker. So that therefore results in a faster sending of the impulse. Does that make sense? I hope that makes sense. And I didn't just embarrass myself. I don't care that I embarrass myself, to be honest. That's the thing. This is what I do. This is how I teach.
Basically, this is me. This is me. Right, I am going to go though because it's 10 o'clock, so it is my bedtime. It is your bedtime too. You need to go and have a restful evening. Get yourselves ready, rearing to go for tomorrow. You are very welcome. I'm glad it helps. And well done on the Oxford offer. That's really awesome. And for those of you who are just joining me, why not come back next Wednesday at 9 o'clock and I will answer any questions that you have. I will also just chat about random stuff because that's what I do. But yeah, thanks for joining me. Thanks for buying my study guides. If you bought my study guides, I do really appreciate that. It's 3:30 a.m. there. What? Where are you? Where are you studying from? It's 3:30 a.m. It's 10 o'clock here. I used to have to really work on that because I tutored students in Budapest a couple of years ago when they were doing their IB, because obviously I teach the IB. And I had to always really think about the time difference, but that was only an hour. Whereas if you're at 3:30, what is that? Oh, wow. Oh, well, congratulations. That's even more impressive with regards to your Oxford interview as well. That's your offer. That's awesome. Right, I really need to go. So good night and have a stop studying, go to bed. Have a good rest, everybody, and I will see you. Oh, thank you, Callum. Thank you so much. That's lovely. Right, I need to go. Stop distracting me. This is what I do. I get so distracted.